The enzymes require a certain amount of energy which is necessary to initiate any kind of chemical reaction which is going to take place. This amount of energy is known as the activation energy or free energy of activation. Generally, this amount of energy is required to initiate the formation of products. In general terms, if we want to understand it, it means that generally an enzyme is present in this condition, and this enzyme tends to follow two pathways for formation of product. One of the theories or a mode of action for the enzyme substrate combination is lock and key model or lock and key hypothesis, according to which an enzyme comes in contact with the substrate over here. Enzyme will add on with the substrate and form the enzyme substrate complex. This enzyme substrate complex or ES complex is the first unit which constitutes two small substrate molecules into it and one larger product molecule, which can be seen over here in the second combination. Here we see that one enzyme is two different kinds of products are formed, and these products are contained within the enzyme. The shape of the enzyme is not changing at all. It is the same structure what it was initially. So what we are trying to understand is the catalytic property of the enzyme is located at the specific regions on the surface. And what are those regions? These curved spaces. Initially, if you see the structure of the enzyme, you can see that there are two curved spaces. Those are the active sites or catalytic centers on which, or small pockets on which the attachment of the substrate will occur. These substrate molecules, when they come and get combined at the active site, they start their activity. And once the initiation of the activity takes place, it helps in formation of the enzyme product complex. Now, enzyme product complex is this complex where green and orange color structures are there, globular structures, along with the enzyme substrate. And finally, we see that the enzyme and the product complex has got dissociated to release the enzyme and the products. One of the example can be of formation of carbonic acid, carbon dioxide and water. They combine together in presence of carbonic anhydrase to form carbonic acid. This reaction takes place in the RBCs, and within a just fraction of seconds, the reaction can occur because of the presence of carbonic anhydrase. So, just the presence of carbonic anhydrase helps in making the process quite rapid. There is another one technique or hypothesis which is known as induced fit hypothesis, according to which there is a transition state in between the substrate and the product, and in this state the old chemical bonds break, atoms assume new positions and new bonds are formed. This transition period is highly unstable and persists for a very short period of time. And like generally what happens is the active site does not have a rigid lock and key formation, but as soon as the substrate comes and combines with the uh, enzyme, it forms a structure which is induced fit, which just fits in the shape, completely fits in with the enzyme. And what we see is that the enzyme pulls atoms from the substrate binds together and leaves as a product. So finally what happens, the detached atoms combine and leave. Enzyme recovers its active sites for reuse. So the same model can be utilized in order to explain both the processes, and they explain it quite well. So the product that fits the last conformation of the active site leaves the enzyme molecule, and then the detached atoms combine and leave the enzyme. So the later, the enzyme is also free, and even the products are also completely free. So this entire reaction is as such that the enzyme has the presence of active catalytic sites, which are sites for attachment for the substrate. As soon as the substrate binds, there is some change going on in the substrate. Change only occurs in the substrate, not in the enzyme. Enzyme retain its conformation as well its activity. Why that there will be formation of enzyme product complex. This is a very unstable state, and after the enzyme product complex, finally the complete formation of product occurs. Product is completely lost or is removed, whereas the enzyme, it retains or is available for the next set of reactions. So the two models or two hypotheses which we discussed was the lock and key hypothesis, and we also discussed the induced Twist hypothesis. The nomenclature of the enzymes, it is also dependent on various factors. The first thing is that there is just addition of a's in order to form the, or in order to name the enzymes, like sucrase. What is the function of sucrase? Sucrase helps in breaking down of the sugar molecules. So lipases, they help in breaking the lipids. Proteineases or proteinases, they help in breaking the protein. Nucleases helps in breaking of the nuclease. So just by addition of a's to the root word of the substance, we can form the enzyme, or we can name the enzyme. Enzymes can also be named according to the kind of reaction they facilitate. For example, or reaction they can catalyze. For example, dehydrogenase, which helps in dehydrogenation reaction. Polymerase helps in polymerization reaction. So these are common examples. According to the kind of activity they are performing, they are named. The name of some enzymes consist of two parts. The first part indicates the substrate, whereas the second part indicates the reaction which is getting catalyzed. For example, glutamate pyruvate transaminase, which transfers an amino group from the substrate glutamate to the substrate pyruvate. So this is one of the examples. Another example can be of DNA polymerase, which helps or catalyzes the synthesis of DNA. So these are some of the ways by which we can name the enzymes. One more technique is there, which means that some enzymes have been named after their sources, like pepin can be extracted from papaya, which is ananas, comosus, and even bromelain can be extracted from another kind of plant, which is pineapple. So, papain from papaya. This has a property of hydrolysis, papain, and even pineapple or ananas. It also releases, or it also has presence of bromelain, which is a kind of hydrolytic enzyme. So these are two of the enzymes, but basically the classification system is as such, over where we use the name of the enzymes and the activity. We focus more on the activity of the enzymes. So classification of enzymes according to the type of reaction they can catalyze. So there are six categories, and we have to learn the functionality, the names, and even one of the examples. First is oxidoreductase. So these enzymes, they catalyze the oxidation as well as reduction reactions by transfer of a hydrogen or electron from one to another. A compound is reduced on accepting a hydrogen or electron and is oxidized on losing of hydrogen atoms and electrons. This is known as oxidation as well as reduction. In oxidation, either oxygen will be added, or removal of hydrogen will be there, or addition of electron will be there. Similarly, in reduction, what happens is, once again, let me just scribble it down. In oxidation, what happens? There are three ways by which we can oxidize a reaction. Addition of oxygen, we can remove a hydrogen, or we can remove an electron. So removal of hydrogen, electron or addition of an oxygen molecule results in oxidation, whereas reduction means removal of oxygen and addition of hydrogen and addition of electrons. So that is reduction reaction. One of the common examples of this kind of reaction is reactions or oxidases. These both are similar kind of reactions. One of the common examples which you will be learning soon will be in photosynthesis, over where 3-phosphoglyceraldehyde dehydrogenase is used in order to convert 3-PGAL, which is 3-phosphoglyceraldehyde. 3-phosphoglyceraldehyde will be converted to 1,3-diphosphoglycerate. And in this 3-PGAL, there is presence of NAD. Now NAD, what will happen to NAD? It will get reduced. It will form NADH. Similarly, there will be addition of a hydrogen ion as well. So this is a proper oxidation reduction reaction, over where oxidation of PGAL is taking place, whereas reduction of NAD plus is occurring. So both the reactions are taking place simultaneously. Next is the transferase. The transferases are certain substances which help in transferring specific group from or specific group. This specific group can be anything, a methyl group or a phosphate group from one substrate to another. For example, transaminase or kinases are examples. So one common example will be of glutamate. When glutamate is added with pyruvate in presence of glutamate pyruvate transaminase, which we have already discussed about. So glutamate pyruvate, and in presence of glutamate pyruvate D transaminase. Glutamate pyruvate transaminase amino group will be added. And what will be the formation? It will form the alanine, because this is the amine group which is getting added, and we will be forming the alpha ketoglutarate. So glutamate is getting added with pyruvate, and in presence of glutamate pyruvate transaminase, it is forming alanine and alpha ketoglutarate. This is our second reaction. Third is hydrolases. Hydrolases, they catalyze the splitting of larger molecules into smaller ones by addition of a water molecule. Like all the digestive enzymes, like the amylases, or generally the amylases or esterases, they all form or follow the same pathway. So what exactly we have to think of? In presence of pancreatic amylase, in the pancreas or in the stomach, it forms maltose. Maltose formation occurs, or isomaltose is formed, or maltose and isomaltose is formed in our stomach by addition of water molecules to the starch or the glycogen. Okay, so this is a hydrolase reaction. Next is isomerase. In the isomerases, we see that the change of a substrate into a related isomeric form by rearrangement of the molecule occurs. For example, it takes place in the phosphohexoisomerase, which changes glucose 6 phosphate to fructose 6 phosphate in glycolysis. This is a very simple reaction which occurs. Glucose 6 phosphate is converted to fructose 6 phosphate. Here the conformational change is only there because 6 number of glucose molecules are only there in case of glucose as well as fructose. Just the conformation is getting changed, just they are forming isomers of each other. And over here there is presence of an enzyme which is known as phosphohexoisomerase. So phosphohexoisomerase helps in conversion of glucose 6 phosphate into fructose 6 phosphate. This is something which we have to remember, and it's a isomerization reaction. Just the rearrangement of molecules is going to take place. Nothing else is occurring at all. So next is lysis, and in lysis we see that the breakage of specific covalent bonds and removal of groups without hydrolysis occurs. For example, histidine decarboxylase splits carbon bonds of histidine and forms carbon dioxide and histamine. So this is one very simple thing which occurs. But one more example can be thought of, that is of aldolases. And in the aldolases, what we see is aldolase splits fructose 1,6 diphosphate into dihydroxyacetone phosphate and 3-phosphoglyceraldehyde in the process of glycolysis. Glycolysis is there in our syllabus, so we can learn this. Right now it's maybe difficult to remember, but later on it becomes easy only because you will be learning all these reactions. Fructose 1,6 diphosphate in presence of aldolase. What does it form? 3-phosphoglycerate as well as dihydroxyacetone phosphate. Both of them, they are interchangeable. This 3-PGL can form dihydroxyacetone phosphate anytime. They are just conformational isomers of each other. So fructose 1,6 diphosphate, which is a 6 carbon molecule, it forms 3 carbon molecules by the help of aldolases. This is a lysis reaction. And the last one is ligases. Ligases means addition or synthesis. So this catalyzes the joining of two substrate molecules by getting energy from the hydrolysis of ATP molecule. Example is of pyruvate, which gets added with carbon dioxide. Pyruvate plus carbon dioxide in presence of ATP, it forms, and the enzyme over here is pyruvate carboxylase. Carboxylase. LASE. It forms oxaloacetate and ADP. So this oxaloacetate is the substance which is formed after addition of pyruvate and carbon dioxide. 4 carbon atom. Oxaloacetate, 3 carbon atom pyruvate plus 1 carbon atom carbon dioxide. So this is the synthesis reaction which occurs for the formation of oxaloacetate. This is a small table which is very helpful, and you can learn it properly. Questions are asked for this entire table. You will get the entire question being asked on the whole table. So it's important to be learned properly.